Okay, I think we are going to go ahead and get started. Thank you everyone for introducing yourselves and putting in the chat where you're from. We can see that we have many individuals from across the globe. Thank you, thank you, thank you. At this time, we're going to go ahead and get started. So good afternoon. On behalf of the FSHD Society, welcome to today's FSHD University. My name is Ashley Ferreira, Director of Education, and I am joined today by a fabulous team by Dr. Nick Johnson, Vice Chair of Research and Associate Professor of Neurology at the Virginia Commonwealth University School of Medicine, as well as members of the Avidity Biosciences team, including Erica Escartin, Rocio Martin, and Dr. Amy Halseth. We're really excited to get going and to hear the exciting updates that they have for us today. But before we get started, a quick reminder, this session will be recorded and made available on our YouTube channel within the next week or so. Also, for today's webinar, while we do encourage participants to please use the Q&A to submit questions, to utilize the chat, to communicate with each other, we would like to let participants know that we have received a number of questions in advance of today's presentation, and we'll ask our presenters these questions first. However, should we have time remaining, we will open up the Q&A. We look forward to today's presentation, which will provide an overview and top-line data from Avidity's FORTITUDE studies, as well as provide exciting updates to our community. Without further ado, I'd like to hand the floor over to Erica Escartin from Avidity, who will kick off today's presentations. Thanks so much, Erica.
Thank you, Ashley, and thank you for hosting us today. And the Avidity team is very excited to be here to present an update from FORTITUDE, a randomized phase 1/2 trial evaluating AOC 1020 Delbrax in adults with FSHD. My name is Erica Escartin. I'm the head of Global Patient Advocacy here at Avidity, and it's nice to see everybody from across the globe. Just a quick disclaimer, this presentation discusses the clinical development program for an investigational product. No conclusions regarding the efficacy or safety can be drawn from this initial preliminary data. This data is also used for educational purposes, and given this data is related to an investigational product, the data cannot be used to make any individual treatment decisions. The data presented today from the phase 1/2 trial in participants living with FSHD is ongoing and will continue to evaluate the safety, tolerability, and pharmacokinetics and efficacy of Delbrax. And Delbrax is an investigational product and has not been approved by regulatory agencies. The safety and efficacy have not yet been established. Next slide, please. We were very excited in last Friday that we celebrated FSHD, World FSHD Day on June 20th. Here are a few members of our Avidity team with the Orange Smiles campaign. And we shared in the excitement with you guys, and we're excited to continue to support the FSHD community, united to find a cure one day. Here at Avidity, our mission is truly everybody in this call. We are grateful for all of the contributions that not only researchers are making, but really the FSHD community, trial participants, their families, caregivers are doing to accelerate the advancement of a potential treatment in FSHD. We have the privilege of really getting to know the community, very closely through either its meetings at Avidity or focus groups or patient councils. And here in this slide, we have Josh and Jared, who came to Avidity a few years ago to play. They're in a band and then play their instruments and really get to know them as individuals, but also really get to understand also their patient journey and their diagnosis. So we're extremely grateful for everybody's courage and also vulnerability to be open and share their stories with us. Next slide, please. At Avidity, we are really committed and truly committed to improving the lives of families impacted by rare diseases, including FSHD, through the development of new drugs and treatments. In this slide, Amy is also a fierce advocate for FSHD, and we also have the great honor and privilege to work with her closely and really also bring in the patient voice, both in and out of Avidity. We recently held some patient groups and patient councils and focus groups to really understand the burden of the disease. And one of the things that has stuck in my mind since that focus group is a quote that was said in the group. And someone described having FSHD as Alice in Wonderland, falling down the rabbit hole, but never really knowing where the bottom is. At Avidity, we take that quote extremely seriously, and we are really looking to change that and change the trajectory of the disease. Next slide, please. So today's agenda, we have the honor of having Dr. Nick Johnson to present the FORTITUDE overview and top-line data. I'm also joined here by my esteemed colleague, Rosia Martin, who's our global head of the FSHD program, and she will talk a little bit about next steps and how we'll move the program together. And then last but not least, we'll leave some time for Q&A session. Next slide. So when we think about a potential therapy, it's really important that we think about it in the context of the patient experience. In this slide, we're showing data from a large survey conducted by the FSHD Society of people living with FSHD, called the Voice of the Patient Report. And really the most prevalent and impactful symptom categories that patients identified were related to the loss of strength, function, upper extremities, mobility, gait, and core function, which then this results in the pain and fatigue that is experienced in FSHD. In the survey, when people were asked what would be short of a cure and what would be important to them, the vast majority mentioned slowing or stopping the loss of muscle function or regaining the strength in their motor function. When people think about the future, these are really the things that we hear most regarding the loss of independence, losing mobility, and ability to walk, and becoming a burden to their family. Next slide, please. So with that, I will pass it on to Dr. Nick Johnson, where he'll talk about the Delbrax top-line data and trial design. Dr. Johnson?
Great. Thanks, and thanks for the introduction. I am very excited to share this super exciting data with everybody on the call today. We can go to the next slide. So this is really a schematic showing you what Delbrax is. It used to be called AOC 1020. I still cannot pronounce the generic name, but fortunate for all of us, I can say Delbrax. So it's an antibody oligonucleotide conjugate, or AOC. We can go to the next slide. And what that is basically is that we have this antibody which binds to the transferrin 1 receptor, or TFR1 receptor, and that allows the oligonucleotide or piece of RNA, artificial piece of RNA, to get into the cells, because your cells won't naturally take up naked RNA by themselves. And so the antibody really does allow for that efficient tissue uptake. And once the artificial RNA or oligonucleotide is inside the cells, it binds to that DUX4 gene and turns it off and prevents the protein expression. Just as a reminder for those of you who are less familiar with this, FSHD is caused by DUX4 overexpression. We know you don't want it, you don't need it, you're not supposed to have it. So this is really designed to get rid of that toxic gene expression. Next slide. Like Erica said at the beginning, really, this is a significant commitment for all of the study participants and their families, as well as my colleagues and our teams, to continue to contribute for this FORTITUDE study and the overall Avidity FSHD program moving forward. Next slide. All right, so these are the data that we're going to talk about. I'll take a few minutes on this slide and point out a few points. So the overall study is 12 months, or about a year in duration. And you can see that there are two different cohorts. There's a 2 milligram per kilogram dose and a 4 milligram per kilogram dose. And then the other thing that I want to point out is that, and this is going to be really important, as I'll come back to it a couple of times, is that the original design had the dosing about every 13 weeks. And you'll see from the data that once the participants transition to the open label extension, the dosing is 2 milligrams per kilogram every 6 weeks. So that's a really different dosing frequency. And hopefully, as you look through the data, you'll see why that was done. So the overall There was a placebo cohort attached to each group, and the overall objective is to look at the safety and tolerability of the ascending doses and the pharmacokinetics. But of course, there's a number of exploratory objectives or ways to see whether or not you can start to get a hint of whether or not this drug is working, including biomarkers, patient and clinician reported outcome measures, and then some clinical measures, which we'll look at as we get going through the slides. Next slide. These are the people who participate in the study. One thing to point out is that, even though randomization is supposed to ensure that the groups are pretty even, in this particular instance, you can see that most of the people who got placebo were male, whereas the treatment groups were by and large pretty evenly distributed. In general, a reasonably aged group. Everyone had FSHD1 within the study. And based on both the FSHD clinical severity score and the D4Z4 repeat number, you can see that this is a pretty moderately affected group of individuals. And that's also consistent with what the average age of first symptom onset is there, which again, pretty even across the groups by and large. Next slide. This is looking at the long-term safety and tolerability, including those participants reporting stuff in the open label extension. One key take home is that there are no discontinuations within the study. Most of the adverse events, or what you might call side effects, were mild to moderate. There was no severe or serious adverse event that was related to the drug. There were some things that happened that just because of life or the disease itself. The most common related adverse events that occurred in greater than 3 participants, one was fatigue, and another one is that the hemoglobin was decreased. There was unrelated severe non-serious adverse event of herpes zoster in one participant, which was not related to the study or the drug. And there were 3 unrelated severe serious adverse events of broken bones in one participant, which really is most likely related to the disease itself and definitely not related to the study or the study drug. Next slide. So, if you have participated in this or other research protocols, you know that people like me love to take muscle biopsies, but it's invasive. It's not, frankly, very much fun, to be honest. There's only a certain number of muscle biopsies that we can take. And so if you really want to get a feel for whether or not you can measure essentially DUX4 or DUX4 activity or the disease-related activity, so you can kind of use those terms. The very best way to do that would be a blood test, right? I mean, it's much easier to do. You can test it much more frequently, and it really does capture all of the muscles, not just the muscle that was biopsied. We're fortunate, and we'll share some of the data in the slides moving forward, that as part of this program, they were able to identify some circulating biomarkers that do provide an early detection of the response to Delbrax treatment. Next slide. Really what that is this protein called KHDC1L, which is a mouthful. CDEX is much easier to say. Again, you'll hear these kind of used interchangeably. This K homology domain containing one like protein is an RNA binding protein. It is involved in both germ cell biology and apoptosis. What DUX4 is supposed to do in normal biology is that it regulates differentiation when we're all embryos at the 4 cell stage. When it's expressed in adults, it causes cell death or what's called apoptosis. So it makes sense, both from a biology standpoint and the biology of DUX4, that this would be a protein that would be turned on by DUX4. Just if you're Googling it, it's hard to find a lot more information about it. It is significantly elevated in people with FSHD compared to healthy volunteer plasma. That's what this graph is showing you. So healthy, you can see it's very low expression. both from the natural history study Resolve. So thank you if you participate in that. And then also in this study, you can see that significantly elevated in people living with FSHD. Next slide. And the good news is not only is it regulated by elevated and regulated by DUX4, but here you can see a treatment response. So the gray graph that you can see here is those in the placebo group. The orange graph is those in the treatment group. And you can see here that the 2 and 4 milligram doses were pooled as they were not significantly different between each other. And you can see that compared to baseline, that there's a significant reduction in the CDEX expression, and that is fairly sustained. One thing to point out is that you'll see that there's this kind of loading period, which is that blue arrow, and you can see that really drives down that CDEX expression. But as the dosing frequency starts to spread out, you can start to see that it trends a bit up back towards the baseline level, which is one of the rationales for this Q6-week dosing or every 6-week dosing. Next slide. In addition, many of you probably at some point in your diagnostic journey had a CK measurement. This is a protein that's leaked from your muscle, particularly in response to muscle damage or even exercise. And here you can see that similar to the CDEX, that you can see that those people treated with Delbrax had a significant reduction in their CK compared to those on placebo. And then it also drifted up as the treatment interval spaced out. Next slide. And also supporting, again, the dosing frequency as well as the treatment response for those participants who were initially on placebo and then rolled over to Delbrax, which is that kind of gray section, which is the open label extension. You can see that those participants also had a reduction in their CK. And in fact, again, with that more frequent dosing, you can see that the CK continued to improve both in a sustained way and a little bit even more than the kind of 9-month visit. So, sustained benefits from the treatment effect and that benefit's best seen at that every 6-week dosing. Next slide. All right, so let's start to talk about some of the exploratory endpoints in terms of strength and function. Next slide. So as Erica pointed out, both on the patient survey and obviously in clinic with me, we know that mobility is a key driver of both disability and really just, a problem within FSHD. One way to measure that is with a 10-meter walk run. This is about 30 feet if you're living in the United States and haven't done your metric conversion. And you can see that compared with placebo, there was a improvement at 12 months with every 13-week dosing in terms of the change from seconds. So that's that kind of gray bar is the placebo, and the orange bar is the Delbrax group. And again, you can see that that improvement at 12 months. Next slide. Very similar idea. This is a timed up and go, which is really a way of measuring both the proximal leg muscles as well as the trunk stability. And again, looking at overall functional mobility. And again, here you can see that the Delbrax group has an improvement compared to placebo in terms of the change in seconds. Next slide. And this is composite quantitative muscle testing. So this is, many of you who've been to clinic are familiar with your neuromuscular neurologist pushing and pulling on your muscles and measuring the strength. That's called manual muscle testing. That has some variation in it and is not nearly as precise as what's called quantitative muscle testing, which is actually using a system that is more precise but really is just a way of measuring strength. And what you can see here is that across 7 different muscle groups, which are shown on the left, there's a quantitative muscle test. And then what's done is it's looking at the percent predicted normal. So in other words, that number is not just the overall strength within, say, the ankle dorsiflexion. It's the overall strength compared to what we would expect to see in healthy controls. So that's what you're seeing here in terms of the change in the QMT percent predicted normal. Again, an improvement in strength compared to placebo in the treated group. Next slide. Here's another way of looking now at upper extremity mobility. A key hallmark of FSHD is the inability to kind of raise your arms over your shoulders. Again, very difficult to quantitate within a study. And so what's done is this thing called the reachable workspace, which is basically looking at how well your arm moves. And here what they're looking at is, if you look at on the left, how well your arm moves across the quadrant 1 and quadrant 3. So if you're trying to do this at home, most of you will be familiar that you have trouble moving across those two spaces. So here what it's looking at is the change in how well you can move your arm basically over your head with a weight in your hand. And you can see, again, improvement on that particular measure compared to placebo in the treated group on the right-hand side of the screen. Next slide. All right, so those are kind of the hardcore functional measures. Here are some selected patient-reported outcome measures. So these are surveys that were filled out by people participating in the clinical trial. One survey that people often use is something called the PROMISE group of surveys. And looking at how individuals participating in the trial rated their physical function, you can see that those on the treatment group rated that as improving compared to declining in the placebo. Likewise, fatigue improving as opposed to declining in the placebo. And then if you use a different patient-reported outcome called the NeuroQoL, again, people in the treated group reported improving upper extremity function compared to placebo. And interesting, and maybe I wouldn't have expected this at the beginning, also improving in sleep disturbance. And maybe that's because of improved mobility overall, or maybe that's reduced pain. It's a little hard to know, but it's interesting to see that that was improving as well. Next slide. All right, a few more here. Here, looking at how people rated their pain and their overall health. Again, you can see improvement in pain as well as overall health compared to placebo. And then the next set of questionnaires is, just a global impression. So people were asked, how severe is your disease? Overall, are you better, worse, or the same? Sorry. And you can see that people rated their improvement in terms of their overall change from baseline, and also improvement in the overall severity of the disease if they were on the treatment group compared to placebo. So, I feel like I'm repeating myself, but if you've gotten anything out of this, you can see that just across the board, overall improvements in the treatment group compared to the placebo. Next slide. And with that, I will pass it on to Rocio Martin.
Thank you, Dr. Johnson, for the wonderful presentation and walking us through the data. I am going to walk through the two ongoing studies that we have to support potential approval across multiple regions. The first one is designed to support accelerated approval in the US. This is the biomarker cohort within FORTITUDE. This cohort completed enrollment earlier this year, and we're expecting the top-line data basically in Q2 of 2026, so a year from now. We are expecting to submit for accelerated approval with the FDA in the second half of 2026. So this study is a 2-to-1 randomization. We enrolled 51 patients to receive Delbrax 2 milligrams per kilogram every 6 weeks compared to placebo. All patients will roll over, if they choose, to the open label extension. So the patients on Delbrax will continue on Delbrax, and the patients on placebo have the opportunity to receive Delbrax. This is overall a 12-month study, and we are enrolling patients with both FSHD1 and FSHD2. And also importantly, we have expanded the inclusion criteria for age to go from 16 years old to 70 years old. The primary endpoint of this study is CDAX, KTC1L. I'm going to give it a try. And this is very important, right? Because that is basically the biomarker endpoint to support the accelerated approval. And as Dr. Johnson walked you through, this is a marker of DAX4 activity, and we can see early responses to treatment with Delbrax. We're also collecting functional endpoints not shown on this slide, but we're also collecting the same functional endpoints that Dr. Johnson has walked you through. We're collecting them on FORTITUDE as well as also quality of life measures. The second study that we have ongoing is a phase 3 study, and this is going to serve as confirmatory study in the US and also will serve for approval in regions outside of the US. This is a much larger study with 200 patients, 1-to-1 randomization to Delbrax, 2 milligrams per kilogram every 6 weeks, compared to placebo. This is a longer study of 18 months, and this is because what we're looking at here as primary and key secondary endpoints are functional measures. All patients will be eligible to go into an open label extension similar to what we had in FORTITUDE. So Delbrax patients can continue on Delbrax, and placebo patients can receive Delbrax. The inclusion criteria also are FSHD1 and FSHD2. We are also having patients from 16 to 70 years of age. And the other aspect is that we are enrolling patients who are able to walk without a cane or without a walker. So it's limited to ambulatory patients for the most part. This is a global study, and we have sites in the US, in Canada, in the UK, and in multiple other European countries, as well as in Japan. As I mentioned earlier, what we're looking at here are functional endpoints. So these are the ones that Dr. Johnson has walked you through: the 10-minute walk-run test, the time up and go, and strength measurements with by QMT. Right now, the primary endpoint of this study is QMT. However, we have built in the flexibility to change the hierarchy of the endpoints as we receive more data from FORTITUDE and also as we get more data from the natural history studies. This is important because we're currently powered basically for the 3 endpoints. So we're very encouraged by the data seen thus far and the ability to show treatment effect across these endpoints. So just to summarize, we are very encouraged by the data from Delbrax thus far. From basically the molecular level to the whole body, we see consistency. And that's what Dr. Johnson was also pointing out, is this consistency across multiple measures. We're seeing reduction in CDAX as an early indication of Delbrax treatment. We're also seeing reduction in CK, and we're also seeing improvement across functional mobility, strength, and quality of life measures when compared to placebo. We know that for our patients, time is muscle, and we're working towards moving Delbrax as fast as we can towards being the first disease-modifying therapy approved. And with that, I'm going to pass it back to Erica.
Thank you, Rocio. And thank you, Dr. Johnson, for your presentation as well. So as mentioned, once again, truly, truly thank you to the study participants, families, investigators, and research teams, both in the FORTITUDE and the FORWARD study. None of this could have really been made possible without your contributions and your enthusiasm and your help to be involved in the trials. Next slide, please. And this is a build. Thank you. So we're very grateful to partner both with FSHD Society and as well partners outside of the U.S. To really connect, learn, and initially take action right as a community. There are many events that the FSHD Society puts together in the U.S. I know that outside the U.S. there are also a couple of events that happen and connections. So we encourage everybody to connect to FSHD Society or local chapters to really get involved in not only connecting with others, but also advancing FSHD disease awareness. Next slide, please. Also, at the same time, we'd love to connect with you. You can see here our QR code and opt-in form. So if you'd love to stay in touch with Avidity and learn about upcoming news and events related to FSHD, we'd love to hear from you. These are also some photos from last year's walk and roll in San Diego. I know that this year we'll be participating in a couple of them throughout the country, and one of them will be in San Diego. So we look forward to seeing you guys there as well. Next slide. And this is my last slide, and this is Avidity. As a whole company, we meet every year together, and every time we meet together, what we call Collaboration Week, we invite patients, caregivers, physicians to come in and share their stories and perspectives so that our internal teams are keenly aware of what is going on in the community, but also really understand, as Rosie mentioned, that time is muscle for us. We're moving as fast as we can with as much urgency as we can to ensure that there's a potential treatment for FSHD for your families and for the community, not only in the US, but outside of the US. So with that, I want to close the webinar, and I guess we'll move on to Q&A.
Yes, thank you so much, Dr. Johnson, Rosie, or Erica, for that amazing presentation. As a reminder to our participants, we have received many questions in advance, so we're going to go ahead and start with those. However, we may have time to open it up to additional questions. And with that in mind, I encourage you to continue submitting your questions in case we are able to get them using the Q&A feature. So the first question that I actually would like to ask is for Rocio, and we received a few questions. Can you explain what accelerated approval means and how it could affect how soon patients can access treatment?
Thank you, Ashley. So accelerated approval path is basically a pathway that the FDA has put in place to essentially approve therapies in areas with high unmet need. And this approval is based off of a biomarker or an early sign of treatment activity, rather than having to wait for a functional endpoint, or in some diseases, for example, in cancer, rather than to wait for survival. So this mechanism allows to provide access to potentially effective therapies much earlier than otherwise it would be. So in our case, if there was no accelerated approval path in place, we would have to wait for the phase 3 study to read out. And as you see, it's an 18-month study, so that the functional endpoints have the time to mature and show a treatment effect. Instead of that, we're able to have this other biomarker cohort, which is based off a biomarker readout with CDAX, and that potentially will provide an earlier approval and an earlier access to treatment to patients. So with that, we're in a position to have a potential BLA filing in the US in the second half of 2026, rather than having to wait for the phase 3 forward study to complete and to read out, which would take much longer time.
Thank you. Will participants in the current trial be eligible to continue treatment through an open label extension?
Yes, absolutely. So patients who are already receiving Delbrax can continue to receive Delbrax in the open label extension, and patients who are on the placebo arm can basically transition into receiving Delbrax in the open label extension.
Wonderful, thank you. The next question I have is for Amy. Amy, we've received a number of questions regarding biomarkers, and we were hoping, can you please explain what KHDC1L is and why it is being used as a biomarker in this trial?
Yeah, so I'm going to say C ducts because I think it's easier for all of us. But C means circulating, right? So this is something we measure in the circulation. But the longer name, the KHDC1L, where this comes from is this is a gene that's in muscle that's regulated by DUX4. So we know that, for example, when we have done work looking at the muscle biopsy and DUX4 activity in muscle, KHDC1L was one of the genes that we were able to measure to get a sense of how much DUX4 activity there was in muscle. And if you remember last year, we shared muscle biopsy data that showed a reduction in DUX4 regulated genes, and KHDC1L was one of those. The advance now that we have this year is that we're now able actually to measure this in plasma. And so we're able to measure this gene, this protein, as a marker of DUX4 activity without having to go into the muscle and measuring it directly in plasma. So what it actually does is a bit of a mystery. It's not a well-characterized protein other than we know it's a target of DUX4. And so whether it's directly involved in the pathophysiology of FSHD or just goes along with all the other bad things that we know DUX4 is doing, we don't really know for sure. But we're doing a lot of work right now to really understand if there are linkages there and also try and understand, like, do the levels of C ducts that we can measure in plasma correlate to functional changes or correlate to disease severity. And all of that work we'll need to do really as part of our accelerated approval pathway, because what we need to show to the FDA is that C ducts is relevant and meaningful and is actually measuring something that's important and that will predict longer term outcomes. So we have a lot of research work that we're doing right now to really better characterize it. But then the idea is that hopefully this will come together in a package then that we'll use to seek accelerated approval.
Thank you. And is CDUX screening widely available or easily accessible at a clinic or at home?
Yeah, no, right now it's a research tool and we'll see where this goes in the future. It's very early days. Right now it's definitely just a research trial that we're using in our studies. Research tool.
Perfect, thank you. The next question is for Dr. Johnson. Dr. Johnson, what kind of improvements are you hoping to see in muscle function, and when would patients likely begin to feel a difference?
Yeah, that's a great question. So far, and this was in the chats too, really what the data shows that Delbrax is slowing disease progression, which is compared to participants who receive placebo and are showing worsening of their FSHD. So you're seeing that gap continue to widen, at least in a short time period. So I think, much more information is going to come out, both in terms of the open label extension and then within the phase 3 trial, because I'm pretty excited to see whether or not dosing more frequently, like every 6 weeks, might actually lead to true improvements in muscle function. So stay tuned and we'll continue to monitor the data as that dosing frequency increases.
Thank you. Actually, so one of the questions that we received in the chat, the data here is based on doses every 13 weeks. Does the Avidity team foresee more promising functional improvements with a more frequent 6-week data?
That's
I
the
mean, I think
question.
you have to wait and see. Yeah, you have to wait and see how the data mature as the open label extension and then the phase 3. But the studies are designed to answer that question.
Perfect, thank you. And another follow-up question. If the treatment proves effective, how long would someone need to stay on it, potentially for life or just a certain period?
Yeah, I think so. Based on what we understand from the biology of FSHD, you would really probably need to keep the DUX4 suppressed over the lifetime to prevent it from worsening. And that's pretty consistent with the data from that FORTITUDE, where you could see that as the dosing got even out to 13 weeks, you could start to see a trend back towards the baseline values. And so it's, what does it mean over 10 years? That's a great question. But I think for now, it probably means remaining on Delbrax long term, or potentially using another treatment that also reduces DUX4 expression if something else is developed in the future.
Thank you, Dr. Johnson. The next question is for Rocio. We've received a number of this question already. Which clinical sites does Avidity plan to include for the upcoming phase 3 trial, and when do you expect recruitment to begin?
So the study has been initiated and is starting in the US, and we will be opening up the remainder regions in basically as soon as we can. So in the US, we basically have more or less the same sites as FORTITUDE study. We have removed a couple and we have added a few others. So we have a bit more differences within FORTITUDE. In Canada, we have added sites. We also have sites in the UK, and then we're going to be also opening sites in multiple European countries. So those ones are Spain, France, Germany, Italy, the Netherlands, and Denmark. And in addition to that, we're also opening up the study in Japan. You will find information on the sites in the website that Erica showed in one of the slides, which is the forwardstudy.com, I believe it is. We also have the ct.gov posting that is going to go up any minute now, basically. So there you can find information on sites as they become available. What I would say is that I realize there's a lot of enthusiasm around the study. There are a lot of sites that have already wait lists. So I understand how that can be disappointing for many patients that want to participate. I would say that all the work that has been done on Resolve, all the work that has been done on Move, natural histories, all of this is helping us in the development of the BRACs tremendously. So there are other ways that you can contribute to research in FSHD, not just with the participation in the Forward study. There are registries. Please, if you are able to participate in registries, natural history, do so, because it's helping us tremendously to really understand the disease burden and really move forward the BRACs.
Thank you. Erica, with that in mind, if someone is interested in participating in a future trial, what can they do now to prepare or qualify?
Yeah, so building on what Rocio said, right? So I think it's one, continue to raise awareness by sharing their personal journey and story, as well as, sharing resources that they have around FSHD. Another important factor is engage with FSHD Society, and outside of the US, especially in Europe, there's FSHD Europe, and then all the different organizations within each of the countries. We can't emphasize enough the enrollment of natural history studies. Those are really important to really help us understand the trajectory, the burden of disease, and really understand, how to advance and move treatments as well. And then, collaborating with researchers and, patient advocacy organizations. Surveys are also patient surveys, really important, and focus groups as well.
Wonderful, thank you. Another question, can patients get access through compassionate use, managed access, or a managed access program?
Sure, I can answer that one as well.
Mhm.
So yes, a managed access program is also sometimes called or referred as an early access or compassionate use. And that is really the mechanism to access the drug outside of the clinical trial before it's approved. We do believe that, wherever it's possible, participation in clinical trial is really the best and preferred route to access investigational medicines. We do share that sense of urgency, and we do take it really very seriously to ensure that we're moving our phase 3 trial as fast as we can, and that we have the results that we need to get the trial approved. So with that, really right now at the moment, we don't have a managed access program at this time, as we're just really focused to generate the data from Forward to get approval.
Thank you, Erica. Rocio, this next question is for you. What are the next steps and timing for Delbrax in pediatric patients?
Thank you for that question, Ashley. And I know that this is top of mind for many. It is also top of mind for us. Our first step into the pediatric population was by lowering the age group in both Fortitude and Forward to 16 years. That's the first step. And we're actively working towards building our plans for pediatric patients in the lower ages. So more to come.
Wonderful, thank you. At this time, we are going to open it up to other Q&A questions, so please continue to use that Q&A function. The next question I'd like to pose to the group, when can we expect a treatment to be available to the public if phase 3 goes well?
We're not in a position to answer that question just now. There's a lot still to be figured out on the timelines for the phase 3.
Beautiful, thank you. Another question that we received surrounding biomarkers. What is the path forward for using biomarkers? At the International Research Congress on FSHD, there were a number of presentations about biomarkers, but what will it take to settle on one and have the confidence to use it over biopsies?
I'll take that. That's a big question. Like formulating my answer. Do I want to take that? Yes, I will. It really is, I think, a accumulation of data that really builds the confidence that what the biomarker is measuring is what's important and what we need to be measuring. So if you think about kind of an ideal biomarker for FSHD, you want it to be something that probably relates to DUX4, right? That's directly driven by DUX4, especially if your therapy that you're testing it with is a DUX4-driven therapy, because you want its levels to be a marker of how much DUX4 activity you have in the body. You want it to be something that's probably higher in FSHD than in healthy controls, and you want its levels to somehow be correlated with disease severity or disease progression, right? So if you think of a biomarker having very low levels, maybe that means that you're sort of in a stable disease progression, you're not getting a lot worse. And then people who have higher levels, if that's really reflecting more DUX4 activity in the way we think of FSHD, that's a lot more disease drive and, disease getting worse a lot more rapidly. So, if you kind of think of those things, it seems like CDUX does meet the bill. There's a lot more work that we need to do and a lot more data. We're doing a lot using natural history data, our own internal data, to try and build those relationships, to try and put that story together. But really, you need that data. Now, if you're thinking about biomarkers sort of in general, right, and thinking about, well, maybe it's a different kind of treatment. Maybe it's a treatment for muscle building, or maybe it's a treatment for reducing inflammation. You would have a different set of biomarkers. But for all of the biomarkers, you would need some kind of totality of data to really put it together so that you understand what is the biomarker actually telling you, and then can you use it for, and this is where it gets really important, regulatory decision making, because the bar is really high of how much data you'd need, how good is the assay, all of those things to be able to use it for regulatory decision making. So I think there will continue to be multiple biomarkers in development because there are multiple pathways and multiple approaches being looked at for FSHD. But we're really excited about CDUX specifically because of the way Delbrax works to target the root cause of FSHD, which is through DUX4. And so we're going to be continuing to do that work over the next year and a half in anticipation of our accelerated approval submission.
I would encourage everyone to listen to the scientific workshop that FSHD Society held with the FDA in early May, because it does break down a lot of what Amy just mentioned, and it was a really well prepared scientific session.
Thank you, Rocio. The next question that I have, I think, is probably one for you. Do you have a sense of when phase 3 enrollment in the United States will be completed?
Yes, so it's again, I think just to take a step back, it's a 200-patient study, and we have multiple countries, and we want to make sure that all the countries and sites that are participating have the opportunity to enroll patients. We're also keeping in mind the potential accelerated approval in the US. So we're going to enroll patients in the US in the earlier months of the study, and then focus enrollment outside of the US, because by the time we get accelerated approval, we should not have any placebo patients in the phase 3 study in the US otherwise. it gets a little bit messy. So we're front loading the US in the first few months and then focusing enrollment ex-US for the remainder of the study.
Thank you. Another question that we received, will you stop collecting data at 18 months or will there be data collected at longer time points, for example, 5 years or a different time frame?
In the OLE, we'll continue to collect data. So the open label extension will continue to collect safety and will continue to collect blood samples as well as functional endpoints.
Thank you. Is there a difference between FSHD1 and FSHD2 regarding the effect of Delbrax that you've seen?
I can take that. So far, just because of who's been enrolled in the trial, we have only enrolled FSHD type 1 patients in cohorts A and B. I think we do have one FSHD2 person who's now been enrolled in the biomarker cohort. So we don't have any data to answer the question, but just thinking about the mechanism of FSHD1 and FSHD2, while they have different ways of getting there, with the end result being just an increase in DUX4 expression in muscle, we believe that Delbrax should work similarly across the two. And that's another thing that, in phase 3, when we have more patients, and we should have more patients with FSHD2, we'll be able to look at that specifically. But just thinking about it from the mechanism of disease, we anticipate that it would work the same across both FSHD1 and 2.
Thank you. Did participants show muscle growth, not just stopping DUX4?
Yeah, we don't have data on that right now. We have the functional data where you look at strength and, overall, it looks like people are maintaining strength. We did collect MRIs in FORTITUDE, and we don't have all of the data yet to be able to analyze that, but that's something we'll be looking at going forward.
Thank you. Is there a circumstance where you might expand the recruitment pool beyond 200 for forward, and what would trigger that expansion if it does happen?
Not at this time.
That's fine. Okay. Thank you. FSHD isn't a linear disease. Would the dosage be stable throughout life or responsive to periods of quicker decline based on what you've seen in the study?
Dr. Johnson, would you like to
Yeah,
address that?
I think it's important for the people listening to realize that this is, a very promising but very small clinical trial. The phase 3 trial will add to that information, but to say that we fully understand how to use this medicine in clinical practice would not be correct. So really, this is going to be a team effort for multiple years, I would say, to continue to explore different dosing, different age groups, et cetera. But I think for now, I would say, the early indications is that it would be that phase 3 dose. And I would expect that at the moment being a pretty stable dose throughout the lifetime, but we'll check back in a few years.
Thank you. Dr. Johnson, for those who aren't completely familiar, can you please give just a little bit of a rundown of how Delbrax is delivered to the patient, whether it's oral, injection?
Yeah, so this is an IV infusion, and it can be, administered in an outpatient. It could be administered in an outpatient setting in the future, I would expect. And it takes a couple hours to infuse it. I mean, depending on how people tolerate it. If you ever had IV infusions for anything else, it would feel very similar to that approach. Is that very similar to other antibody drugs that we use.
Thank you. This might be a question for Eric or Rocio. How can I ask my doctor to approach nearest trial sites for Global 3 phase? I'm interested in participating.
So you want me to take that? Yeah, the best way is we've shared the link to the forward study. So the best way is to go to that website, look at the list of sites, and then see what's the closest nearest site to your location.
Thank you.
And maybe just to
Another
chime in that
piece.
as Rocio mentioned, most of those sites do have waiting lists to begin with. So please be patient as one of the study sites contacting us. I think we're all anxious to get the study enrolled as quickly as possible, but it does take a little while.
Thank you. Yes. Another question that came in. So is the use of artificial intelligence offering opportunities to help testing or approval or at all impacting research?
Yes, so absolutely, and we're very happy to have a AI expert within Avidity, and definitely we're making a lot of use to expedite as much as possible all the processes and writing documents, etc. So we're leveraging AI.
Beautiful, thank you. Was there a solution for the anemia fatigue which affected patients in FORTITUDE? Will this subside with the new dosing?
Amy, why do you want to comment on the differences between the dosing frequencies or the amounts?
Yeah, so going forward in both the biomarker cohort and in phase 3, we're using 2 milligrams per kilogram given every 6 weeks. And what I can say, we haven't unblinded any of the data in cohort C yet, so we can't say for sure, who's on active or not. But so far, it's seeming like patients are doing okay in that cohort, and we'll just have to wait for the data to all mature to really know for sure.
Thank you. Did you collect data on DUX4 activity from the dose escalation? And if so, why was it not shown?
So we have looked at DUX4 activity in muscle biopsies and both the 2 and the 4 milligram per kilogram dose. And we've also have all that functional data that was shown. And just for the sake of time, we've not presented the data by individual dosing. But what I can say is both in terms of CDUX, as well as the muscle biopsies, as well as the functional endpoints, we don't really see a difference between the 2 and 4 milligram per kilogram in terms of any of those endpoints. So we think what that means is if you think of a dose response curve, at a certain point, you've kind of maxed out your dose response curve, and going higher doesn't get you any extra activity. So that's sort of how we're envisioning it. And that's why we've chosen to move forward with this 2 milligram per kilogram dose and then giving it more frequently, as Dr. Johnson mentioned. When you look at CDUX specifically over the longer dosing interval, it looks like things are maybe ticking up a little bit and returning toward baseline. So we think that keeping it every 6 weeks will allow us to keep DUX4 suppressed over the entire dosing interval.
Thank you. Will there be a baseline CDUX inclusion requirement?
No, we'll measure it because, of course, we want to look at changes from baseline, but there's no entry level of CDUX that you need to have to get into the trial.
Wonderful. We are just right about time, so I think that we'll go ahead and close the Q&A at that point. Thank you, Dr. Johnson, Erica, Amy, so much for being here and for presenting this information to our community and for answering all of the questions to our community. Thank you so much for sending in all of your questions. They show just how engaged you are. Amy, did you have something that you wanted to share?
No, thank you. I was just looking at the comments.
Okay. Yes, okay. You look like you were ready to jump in there, but no, thank you again to our presenters. We really appreciate it, and to all of you for being here. I would like to remind you that our next FSHD University will be this coming Thursday, and we do encourage you to attend. It will not be at our usual time, but it will be Breathing with FSHD, taking place Thursday, June 26th at 5 p.m. Eastern. We also encourage you, of course, to look at our calendar events for other upcoming events in your area. And again, thank you for joining us, and we'll see you again soon. Bye, everyone.
Thank you. Bye.